Plastic pollution is one of the leading sources of ecosystem degradation. Macroplastics are characterized as pieces of plastic bigger than 5 millimeters and are visible to the naked eye. They can be removed and cleaned up easily, but if they are not, they can stay in nature for years or even centuries. As macroplastics fragment into smaller pieces, they enter food chains and affect all different ecosystems. Despite the understanding of where plastic pollution originates from, a knowledge gap remains concerning the effects of macroplastic on coastal plants along the inundation gradient. Due to the gap, the ability to fully assess the long-term effects on the environment is limited. Addressing this knowledge gap helps the environmental and ecological conservation of diverse habitats such as the coast of the Wadden Sea. This study will focus on the effects of macroplastics on the Mediterranean sea lavender and glasswort, which are coastal plants that grow along the inundation gradients, specifically in the Slufter on the island of Texel.
Our research question is, what is the effect of macroplastics on the survival rate and success of sea lavender and glasswort over the inundation gradient? We thought of two specific research questions that will help us answer this question. For each SRQ, we composed an hypothesis of what we think will be the end result of each experiment we design.
For our experiment, we used a device called the PAR fluor pen. PAR stands for photosynthetically released radiation, and the device measures it in ppfd, photosynthetic photon flux density. The higher the ppfd value is, the higher the amount of usable light the plant receives. Before measuring the leaf using the PAR fluor pen, we first had to dark depth the leaves that we were going to use the device on. We did this 60 minutes before doing the measurement on all plants in both the control group and the experimental group. The plant we were doing this measurement on was the Mediterranean sea lavender. We collected this plant at the Slufter using syringes. We got 16 plants so that both the control group and the experimental group had 8 plants. We put them in 2 of the same containers. We used the PAR fluor pen on the control group and the experimental group. The experimental group will have a trash bag over their container and the control group will be open.
Secondly, we did our roof strength test. For this experiment, we took measurements of both the Mediterranean sea lavender and the glasswort plant found in the Slufter on Texel. We did a pulling test using an analog force gauge. First, we calibrated to 0 and then hold it horizontally. By attaching the hook of this device to the base of the plant and pulling it apart until it breaks off, using both the device and our own force, it measures the strength of the root. We measure the strength of the experiment and the control group of the photosynthesis experiment.
For the data analysis, we put all gathered data in Excel. We organized the findings in tables and visualized them with graphs for comparison. The different experiments that we did help answer different parts of the research question. The PAR experiment answers the part related to the effect on plant functioning. We compared the average rate of photosynthesis in control groups with the plants that were covered. This way we could see if macroplastics are linked to reduced photosynthesis. We used an independent samples t-test for this experiment. If the p-value is lower than 0.05, we reject the null hypothesis, meaning that there is a significant difference between the groups. We used the root strength experiment to see the effect on plant survival. We compared the average root strength of both species from various inundation levels and also between the covered plants and the control groups. Weaker roots may indicate lower resistance and thus survival potential. Moving on to the results of our experiment. Our first hypothesis stated that macroplastics cause lower rates of photosynthesis. We made this table to organize our findings and create graphs. As you can see in this graph, the ppfd of the control group is significantly higher than of the test group. For our last measurement, we did a t-test, as shown in this bar chart, of the average of the measurements which gave a p-value less than 0.05. This means that there is a significant difference between the two groups. Our second hypothesis stated that plants with weaker roots will break more easily due to the macroplastics. As you can see in this table, there is little to no difference between the control and test group. Based on these results, we cannot conclude whether macroplastics have any effect on the root strength of plants.
This study investigated the effect of microplastics on coastal plants in the Slufter on Texel. The shading experiment showed that plants covered by plastic received significantly less usable light than the control plants. All t-test p-values were below 0.05. This supports the hypothesis that microplastics may negatively affect plant functioning by reducing the light for photosynthesis. However, the experiment did not measure the actual photosynthesis rate, the growth or long-term survival of the plants. The plastic covering was also a simplified simulation of microplastic pollution, and differences in weather and time may have influenced the measurements. The tested plants sea lavender and glasswort are also very resilient. Therefore, the experiment was on the short side of only 4 days to notice change. The root strength experiment did not clearly show the microplastics weakened Mediterranean sea lavender or glasswort. The force gauge measurements were limited because the hook sometimes damaged the plant above the root instead of measuring the strength of the root itself. Or the root was too small for the hook to even put in. Overall, the results suggest that microplastics may negatively affect coastal plants through shading, but further research with more plants, longer exposure times and improved methods is needed.